Invite Dr. Tanay Bhat for the next presentation. Tanay is a co-founder and CEO of Koshkey Biosciences, and he is my colleague in National Center for Biological Sciences, and now he's working as a startup focusing on antimicrobial resistant AMR and wound healing solution. Tanay, the stage is yours.
Thank you. Thank you very much, organizers, and Bhuvna for inviting me over here. As she mentioned, my name is Tanay Bhat, and I co-founded Koshkey Sciences along with Professor Colin Jamora about 2.5 years ago. In Koshkey, the Kosh means in Sanskrit a cell, a biological cells. We are all made up of multiple billions of cells. A single cell is called Kosh, and key is to unlock the potential of that cell. So the vision behind Koshkey is that can we use our body's own cells and unlock their potential to become disease-free? But it's, of course, easier said than done. So what we are trying to do is bring in novel RNA interference therapies for various kinds of diseases. And in this presentation, I'll take you through how we are doing it. But to begin with, let us understand what is the problem. The problem is that there are more than 10,000 diseases for which there is no treatment. These diseases are the ones which are rare and genetic in nature. And because they are rare, many of these diseases, we are not hearing them every day. What we hear every day is probably cancer, diabetes, and various kinds of resistant infections. And worldwide, they are the reasons behind, hundreds of deaths. However, the other rare genetic disorders which are not treatable are mostly treated, I would say, only for the symptoms. The current medicine is not treating the root cause of it, and hence patients are often frustrated because they have to keep on taking these medicines lifelong, and eventually, if you consider the cost they are spending for these therapies, it's a lot. So most of these diseases, as I mentioned, are genetic in nature, and if you want to treat a genetic disease, what would you do? And as the previous talk suggested, you would have to go for the genetic therapy. CRISPR-Cas9 is one of the ways you can do it, but in the umbrella of gene therapy, when you want to change a DNA code, and that too in each and every cell of your body, it's a very, very huge task, very difficult task. And traditional gene therapy that's why very rarely we see in the market, very rarely we see developed within India and given to the Indian patients. And that's why we need a new solution. What are the problems with the genetic therapy currently? It's a very high cost of investment. At least $2 billion you have to spend to bring out one therapy to the market. Second problem, high risk of failure. Only 28% chance that whatever you're starting today, after 5 years, may reach to the market if it is into this genetic therapy domain. And the third is because you are spending so much time, energy, and investment of various investors, you'll have to charge patients a lot. And that's why you might have heard that a single injections for the DMD, Duchenne muscular dystrophy, which is one of the rare genetic disorder, cost more than $3 million per patient. Of course, that is a very, very small population of patients which can afford that. And hence, these kind of therapies are not becoming very popular, not only in India, but elsewhere in the world as well. So what is the solution? We are bringing in RNA interference therapy, which addresses key obstacles faced by the traditional gene therapy. What is the meaning of RNAi therapy? It's very simple. We are interfering with mRNA of a particular gene. It will lead to gene silencing of that particular gene which was causing the disease. And it's a solution for the long-term disease resolution at an affordable cost. So very briefly, I'll go through how this entire therapy works. And it will be a sort of an oversimplification because I'm considering that, most of the students right now might not know everything in detail. But as I mentioned, that it all starts with a mutation in your DNA. Because your DNA has a mutation, your RNA gets a mutation, and because of that, that unwanted protein gets produced, which causes the disease. Now, what you're seeing in the red color is antisense oligo, which is precisely tailor-made for that mRNA. We are not right now altering the DNA, but rather designing antisense oligo which will bind only to that mutated mRNA and degrade it. This antisense oligo will result in the silencing of that gene, no protein production, and hence no disease. When we compare these RNA interference therapies with the traditional gene therapies, there are several advantages as well as it's comparable. It has, you can target, in fact, any gene in the body because each gene and each mRNA has its own sequence, its own signature. It has high efficacy and it is long lasting. You can administer this therapy not only through the veins or subcutaneously, you can also inhale it. It can be applied across multiple areas, not only rare genetic, but nowadays these therapies are coming up for various commonly occurring diseases as well, such as hypercholesterolemia, heart diseases, kidney diseases, brain diseases. The cost of development, that's the most important thing for any pharmaceutical company. The cost of development is low, hence the cost of therapy to the patient is low. And I would say on the humanitarian side, the safety of this therapy is also very high. There are very low safety issues you would face in RNAi therapies. Why? Because if in the gene therapy something goes wrong, that something goes wrong will be embedded in your DNA for the rest of your life. But in the mRNA therapy, your DNA remains as it is. You are only targeting the mRNA, which is continuously being produced. And this therapy is, something which is not going to be integrated in your DNA. Hence, it is having very low safety issues. So our mission is that can we make this indigenously affordable, low regulatory hurdle RNAi therapies for diseases with high unmet need, for which often they are called orphan diseases. No drug is existing. And can we support research for the same for various other companies as well? Right. So what we have done over here. So of the many things which are in the pipeline, I will be explaining just one of the therapies, which is targeting the AMR. Why AMR? Because if you look at various other diseases, AMR currently might look small, but by 2050, we will be seeing that every year, 10 million people will be dying because of the antibiotic resistance. Why is that happening? Of course, because we are, particularly in India, antibiotic use is uncontrolled. Bacteria continuously are evolving. So today, whatever antibiotic is working for a bacteria, tomorrow it might not work because that bacteria has evolved against it. The number of deaths you see might become higher than the number of deaths which is happening because of the cancer. What we have also observed that the antibiotic resistance does not affect all of us equally, rather certain population, those who are undergoing chemotherapy, those who are having HIV, or those who are diabetic are more likely to get these kind of infections. So there is a strong correlation between those population having weaker immunity are also going to get various kinds of infection. And if you think about AMR infections, they are often hospital-borne in nature. People with lower immunity get it, and people who are having non-healing wounds, those wounds often get it. So we know that if you are a healthy individual, if you get any injury, you do not worry much. Injuries are part and parcel of the life. There is nobody who can say that I have never got injured. And we don't worry much because it often gets healed nicely. But if we are a diabetic individual, our skin cells, particularly called skin keratinocytes, do not sense this injury, and it leads to progressive diabetic foot ulcers. This kind of foot ulcers, after a certain stage, also attracts various kinds of infections, necrosis, and after certain points, it's non-returnable. It leads to either limb amputation or sepsis-mediated death of that individual. So how can we intervene this particular program? So what we discovered as part of my PhD program, I was working on it, that every skin cells have a pool of AMPs, which are called antimicrobial peptides. So if I take any bacteria and put on the skin of a healthy individual, mostly nothing happens because our antimicrobial peptides take care of it. However, in the diabetic individual, there is a break on the release of these antimicrobial peptides. We will call it for now a break gene. And when the break gene is highly expressed, the pathogen cannot be neutralized and it will continue to grow on your skin. So what do? We design a therapy, siRNA, small interfering RNA. You make it a paste or ointment out of it and deliver this topically near to the diabetic wound. This siRNA goes inside, silences that break gene, and once the break gene is gone, automatically antimicrobial peptide secretion starts. These antimicrobial peptides will also kill your pathogen neutralization, as well as it will kickstart the wound healing program. So in one stone, you are killing two birds. You are also neutralizing the pathogens which are AMR in nature. You are also increasing the speed of wound healing, a perfect solution for a diabetic non-healing wound. So this is what I summarize, that if a patient is coming up with a chronic non-healing wound, the Koshkey's therapy KK03 will be applied topically, which can bring back the healthy healing. It can resolve the infection as well as bring back healthy healing. This is how the therapy would look like in future. It would be topical in nature, antimicrobial peptide mediated neutralization of pathogen as well as faster wound healing. How did we come up with this thing? This is not just a hypothesis. We had to work on it at in vitro level. We took the cells of a diabetic population, we grew them in the culture, we observed that this was happening. We tried these in vitro conditions on the human cells, as well as we developed a mouse model of diabetes. This diabetes mouse model, just like a human patient, also shows this defect. And we tried to treat these diabetic mice with this particular formulation. So what you are seeing is that in this particular graph, if your mouse is wild type, which means no mutation, just like a healthy human being, a blue line shows that a particular size of a wound will heal in about 12 days. But if the mouse is diabetic, just like diabetic human being, it will take 24 days. If you convert that 24 days into the human population, it would turn into some 3 months or more. But now when you're applying RNAi, the green line, you would see that 24 days becomes 14 days. And we have to understand that what it means for the human diabetic population. It would mean that it's a 40% faster wound healing, 12 times more effective against infections, 30 days reduction in the follow-up period, and 25% reduction in the total treatment expenditure and opportunity cost. So moving forward, I just want to introduce you that when you are interested in developing any new drug, it is not a simple stages of moving from idea all the way to market. There are multiple stages you will have to cross. For this particular drug, what I am describing, it all started with the basic research, where we identified that one break gene was the gene which was causing issues in the diabetic population. That's where it's called idea stage, a target discovery stage. We worked out that in Institute for Stem Cell Science and Regenerative Medicine, as well as part of the research happened in Milan, IFOM Molecular Oncology Institute. Then we did the target validation, that whatever we are thinking that this medicine will work, will it really work? So we had to try it out in vitro condition in the human cells, explants, as well as the diabetic mouse model. That's target validation and lead generation. And currently we are in the efficacy and safety stage. You will have to convince the regulatory agencies in any country that this particular drug is also safe enough to be applied in the human. After that, you get early phase clinical trial approval. That's called phase 1 trial, which will be followed by phase 2 and 3. And then if you are all lucky, everything efficacious and safe in the human as well, then you reach the market. So we are currently in the preclinical development stage. So with that, I'm thankful for all of you. And if there are any questions, maybe now or later on, we can meet and discuss. Thank you.